Okay, folks, so I'm going to introduce you to some challenging material today. And this is going to seem like probably the hardest stuff we do at the beginning of this class, but I'm going to provide you 3 steps to follow that should always lead you to the right answer. So please make sure you write this down clearly so you can follow it when I give you examples of this in the future on homeworks. So, since we'll be talking a lot in this class about amino acids and their functions, their interactions, we need to understand what happens to amino acids when they're around other stuff, right? Because our surroundings affect us, and that's true for amino acids as well. So the pKa's of ionizable groups, so the pKa's of anything that can gain or lose a proton, in particular, the R group is what we're going to focus on here. Why are we focusing in on the R groups, folks? Well, if I take us back just one slide to discussion of peptides, you'll see that in a peptide or in a protein, really all there is are the R groups. Because for any peptide, there's only a single N terminus and a single C terminus that is charged and can gain and lose electrons. Gain and lose protons. Since there's only one N and C terminus that can gain or lose a proton, we need to then focus in on the R groups, because the R groups then are what's going to be doing a lot of the function. And in this example, we are going to talk about histidine. So here is the amino acid histidine. I'm going to fix my picture a little bit by adding in that lone pair. The R group of histidine is what I'm showing you because we are going to be ignoring the N and C terminus. Remember, in a peptide, the N and C terminus do not gain and lose a proton anymore. So what might happen to histidine if we put it in an environment where we have something charged? Will histidine be more likely to gain a proton? More likely to lose a proton? Let's see, and then let's determine what happens to histidine's pKa when we switch up its environment. Okay, folks, here comes the 3-step process. Write this down, pay attention, because it will help you solve these problems. So in this example, what we're going to do is we're going to have histidine in the environment of negatively charged carboxylates. So, a 3-step process. Step 1. Draw the amino acid, both protonated and deprotonated. Step 1. Our amino acid is histidine. So I'm going to draw out this R group, both protonated and deprotonated. So first, histidine without its proton, and now histidine with its proton. So that's step 1. I drew histidine both protonated and deprotonated. Step 2, draw in the environment. The environment is going to be a negatively charged carboxylate. So I'll just draw the functional group of interest. There is the environment drawn in. Final step, step 3. And this is the hard part. Draw a conclusion. And this is the hard part, because we have to use the knowledge that we have already in this class to decide which is more favorable. Okay, so let's walk our way through it. Let's start on the left-hand side of my screen. So on the left-hand side of my screen, we have histidine. And I'm just kind of going to write some things down along the way to help us keep track of what we discuss. On the left-hand side, we have histidine. As histidine is drawn, we would describe it as polar because we have nitrogens and hydrogens that have polar bonds and will have partial charges. So I would describe histidine as polar. Histidine is interacting with this negatively charged carboxylate. So for sure, the carboxylate is charged. So one thing polar interacting with one thing charged. What is the name of the interaction between one thing that is polar and one thing that is charged? This is something I want us to be very comfortable with in this class, is naming interactions that we see. The interaction between one polar and one charged thing is called ion dipole. So that is kind of an analysis of what we see on the left. Now let's look at the right-hand side scenario, starting with histidine. Histidine is protonated, so it is charged. The environment did not change, it is charged. What do we call the interaction between two things that are fully charged? That is ionic, okay? Now I'm going to draw in our knowledge of Coulomb's law real quick to ask us, is this a favorable interaction or an unfavorable interaction? Do we have attraction or repulsion? Well, here we have the interaction of oppositely charged species. So this is a favorable, attractive interaction. Finally, folks, we're supposed to be drawing a conclusion about which scenario is more favorable. Well, what's stronger? An ion dipole interaction or a favorable ionic interaction? Ionic is stronger than ion dipole. So, check. This right-hand side scenario is more favorable, and the left-hand side scenario is less favorable because of the strength of the interactions present. Ionic interactions stronger than ion dipole interactions. Of course, we had to check ourselves with Coulomb's law and ask, is it an attractive interaction? Because if we had a positive and a positive that repelled each other, we have to recognize that is unfavorable. So here's your three-step process to answering the question, how do we determine environment effects on R group pKa? Just got to take the last little step forward. If we determined the right-hand side scenario was favorable, the right-hand side is protonated histidine, right? On the left-hand side, histidine is deprotonated with a lone pair. On the right-hand side, histidine is protonated. So if histidine has its proton, it has taken a proton, histidine behaved as a what? If histidine has accepted or grabbed onto a proton, has it behaved as an acid or a base? Well, bases accept protons. So we would say that histidine behaved base, or like a base, or more basic. If it is behaving more basic, its pKa should be more basic as well. So we would expect the pKa to increase. If histidine is behaving more basically, its pKa should increase. Let's do a quick recap, folks, because I know this is tough stuff. We did a 3-step process. Step 1, draw the amino acid R group of interest, both protonated and deprotonated. Step 2, draw in the environment in the question. Step 3, draw a conclusion about which scenario is more favorable, protonated or deprotonated. We determined the protonated version in this case was more favorable because that allowed histidine, being positively charged, to have a strong, favorable ionic interaction with negatively charged carboxylate. We know opposites attract through Coulomb's law. And this ionic interaction is better than the ion dipole interaction if histidine were to be deprotonated. Since histidine kept its proton and behaved as a base, that means its pKa would increase because it should be more likely to keep its proton and keep that positive charge if it's hanging around negatively charged things. And that's your 3-step process, folks. It will help you come to a conclusion in every single question I might ask you that is similar to this. One thing I need to point out to you, folks. In this scenario, I did not perform acid-base chemistry between my R group and the environment. In these examples, folks, the environment does not gain or lose protons. The proton that histidine took, let's just assume it was from a water somewhere. So when you're answering these questions, please make sure that your environment is not the one gaining or losing a proton. Do not do chemistry between this R group and its environment. That wraps up our videos for today, folks. We better know our amino acids by heart for this learning check. Long term, you won't need to keep them memorized, but I think it's nice early on to get them into our brains because we talk about amino acids so much in this class. It would be great if you had some quick recall. If I were to say, oh, arginine, you can see it in your mind. Oh, serine, you can see it in your mind, right? It's also going to be important for us to know amino acid structure, polarity, charge, because these things will be very important to their function as we move through this class. We should also start to be able to predict how an environment an amino acid is in can change its chemical properties. We just saw that if in a negatively charged environment, histidine's pKa is probably going to increase. Thanks so much, folks. I'll leave you with this cat meme. See you in class.